When it comes to the different products that were simply created by the light-dependent reaction, which are the NADPH and the adenosine triphosphate, both of these materials will now be utilized on the Calvin cycle. Calvin cycle, it is known as the phase 2, also known as the dark reactions or the light-independent reactions, and it is now happening in the stroma. Remember, the light-dependent reaction is happening on the thylakoid membranes, while in the case of the dark reaction or the Calvin cycle, it will now happen on the stroma, in which it is actually possessing three major phases, which are carbon fixation, reduction, and regeneration. As a summary of the different things that you're actually going to consider. So for phase 1, it's known as the carbon fixation, or just the attachment of the carbon dioxide that is from the atmosphere, obviously, a 5-carbon compound known as RUBP. Then, it will create a short-lived intermediate. Then, it will now create 3-phosphoglycerate. Then, with the presence of adenosine triphosphate, it will now create 1,3-bisphosphoglycerate. Then with the presence of NADPH and the release of phosphates, it will now create the glyceraldehyde triphosphate, or also known as the G3P, and some of this glyceraldehyde triphosphate will be released to create now the food material known as glucose, while some of them will undergo the process of regeneration to recreate now the RUBP, which will be responsible for the continuation of the cycle. Okay, so now, what are actually the different things that we are going to consider when it comes to this particular process? When it comes to the phenomenon known as the Calvin cycle, we're actually going to start our discussion here with the presence of a 5-carbon compound known as the ribulose 1,5-bisphosphate, or also known as the RUBP. The ribulose 1,5-bisphosphate, as you can observe here, from the name itself, it is a 5-carbon sugar, then 1,5-bisphosphate because it is possessing phosphate on carbon number 1 and carbon number 5. That is the reason why it is named as ribulose 1,5-bisphosphate. Okay? And in the case of the ribulose 1,5-bisphosphate, this would be the location in which the carbon dioxide will enter and will attach itself. Okay, so now let's consider Calvin cycle utilizing coins. Okay, for me to properly discuss to you how Calvin cycle is actually happening, let's represent every carbon backbone with coin. In the case of carbon fixation, remember it is just the presence of the RUBP, or the ribulose 1,5-bisphosphate, which is a 5-carbon compound possessing a phosphate group on carbon number 1 and carbon number 5. Then, the presence of carbon dioxide, since it is not possessing any types of phosphate, I'm not going to indicate it because the phosphate is more important than oxygen and hydrogen when it comes to this certain type of representation. So in the case of the carbon fixation, this would be the very first step. It is in between the RUBP and the carbon dioxide. Then, the next thing that will simply happen is that once the RUBP and the carbon dioxide combines, they will now create here the substance that is known as a short-lived 6-carbon intermediate. Originally, this would be what a 6-carbon compound would look like. But in the case of the process of carbon fixation, since there are actually no available space here, the carbon that is from the carbon dioxide will actually attach on carbon number 2 of the RUBP. So this is now known as the short-lived 6-carbon intermediate. Why is it on carbon number 2? Since there is only the available space after the enzyme known as the Rubisco has done its job, or The rubisco is the enzyme that is responsible for the attachment of the carbon dioxide to the RUBP. Rubisco stands for the ribulose 1,5-bisphosphate carboxylase oxidase. In the case of the carbon fixation, the very last stage is when the short-lived intermediate 6-carbon compound is simply cut into half. Okay. The last part of the process of the carbon fixation, it is now when the short-lived 6-carbon intermediate compound is simply cut into half to create now 2 3-carbon compounds which are possessing one phosphate on each end. And this substance is known as a 3-phosphoglycerate. Okay, so now, why is it named as phosphoglycerate? Remember, if you're going to consider your chapter 2, which is all about the presence of biomolecules, 3-carbon compounds, they are the glycerol backbone, because they are possessing phosphorus on the glycerol backbone. Then 3 because it is now the carbon attachment. So remember, whenever we're talking about here, in this particular case, sir, this is carbon number 1, isn't it? But remember, whenever we are talking about this per se, when it comes to the chemical naming, we are considering this as the 3rd carbon, and this is also the 3rd carbon, since they came from a single type of compound. 3-PG, or the PGA, or also known as the 3-phosphoglycerate. 3 carbon backbones, then the presence of the phosphate group. In this case, it is present on the 1st carbon, but this is still named as 3-PGA. I won't complicate things when it comes to this particular matter, since this would be discussed to you when it comes to the nomenclature in chemistry. But remember, when it comes to this particular process, we are still going to name them as 3GP or PGA or 3-phosphoglycerate. Okay? This would be carbon number 1 to carbon number 3. Then, even if we are going to invert it, this would still be carbon number 1, carbon number 2, carbon number 3. Why? Since the double bond to oxygen is present on carbon number 1. This is an example of a 3-phosphoglycerate. Okay, so this is the end of the first stage, known as the carbon fixation. Now, for the second stage that is present on the Dart reaction, it is now known as the reduction. When it comes to the reduction, we're actually going to start with the presence of the 3-phosphoglycerate. The next thing that will simply happen to them is that the 3-PG will actually have interaction with adenosine triphosphate. Okay? And when adenosine triphosphate will enter, it will now release the 3rd phosphate that is present in this ATP, releasing now adenosine diphosphate, and the phosphorus will actually be attached to the 3-PG or the 3-phosphoglycerate, creating now a new compound known as 1,3-bisphosphoglycerate. From the name itself, 1,3-bisphosphoglycerate, because it is happening among plants. Bisphospho because they are possessing 2 phosphate groups. Phosphate on carbon number 1 and on carbon number 3. Okay, so from this point forward, we are actually going to have this certain type of process times 2. So in this case, there is now the presence of 2,3-PG, then the presence of 2-adenosine triphosphate, then the release of 2-adenosine diphosphate, and the creation of 2,1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate are also known as 1,3-BPGA. This would be their chemical structure. Once again, carbon number 1, 2, 3. Okay, because it is possessing the glycerol backbone. Phosphates on carbon number 1 and carbon number 3. So it is named as 1,3-bisphosphoglycerate. Okay, so that is the first step when it comes to the process known as the reduction. Okay, after the process of reduction, the 1,3-BPGA will now interact with the NADPH. Once again, where did this NADPH come from? It is from the light-dependent reaction. The NADPH will actually release now hydrogen ions. The hydrogen ions that is present on the first light-dependent reaction. Okay? It will now be released, and with the release of this hydrogen ions, the phosphates that are actually present on the 1,3-bisphosphoglycerate will also be released. Okay? In which only one phosphate group will remain, and that is present on carbon number 3. This is the only phosphate that will remain on this carbon backbones, in which the NADPH will now be once again charged as NADP+. Okay? So, if we have here the presence of two 1,3-bisphosphoglycerate, two nicotinamide adenine dinucleotide phosphate, which is charged, will create 2 NADP+, and it will now permit the release of 2 phosphates, and it will now create a new byproduct known as 2-glyceraldehyde phosphate. Glyceraldehyde 3-phosphate, glyceraldehyde, glycerol, aldehyde functional group, then carbon number 3, there's the presence of the phosphate group. This is now the G3P that will be utilized on the next part of the Calvin cycle. Okay, so just a short recap once again. The very first phase, it is now the presence of carbon fixation. We have the presence of the RUBP plus carbon dioxide, which will now create the short-lived 6-carbon intermediate. Then, it will simply be split to 2,3-PG or 3-phosphoglycerate. Then, the 2,3-phosphoglycerate, it will now interact with 2-adenosine triphosphate, and 2-adenosine diphosphate will be released, which will now create 2,1,3-BPGA. Now, in the case of the 1,3-BPGA, it will now interact with 2-NADPH, 2-NADP+, and will release 2 phosphates, and will now create 2-G3P or glyceraldehyde 3-phosphate. Remember this, the presence of the first part, which is now the carbon fixation, and the second part, which is now known as the reduction. Before we proceed with regeneration, you should bear in mind that regeneration will only happen if 3 RUBPs and 3 carbon dioxide will be utilized. Okay? Now, this would be the case. 3 RUBP plus 3 carbon dioxide will create how many short-lived 6-carbon intermediate? It will be 3 molecules as well. Now, how many 3-PGs or 3-phosphoglycerate will be created? It will now be 3 times 2, it will create 6. Then, how many adenosine triphosphate will be utilized? It will be equivalent also to 6 to create 6 adenosine diphosphate. Then, how many 1,3-bisphosphoglycerate will be created? It would be also equivalent to 6. Then, how many NADPH will be utilized? 6. How many NADP plus will be created? 6. How many phosphates will be released? It is now 6. How many G3Ps will be created? It is also equivalent to 6. That's the case whenever we are talking about the Calvin cycle. Now, before we proceed with the last step, which is regeneration, we are going to release first 1 G3P. Okay? 1 G3P will be stored on the stroma while 5 will return to create and to do the process of regeneration. Okay? 1 will be released, 5 will be reutilized. Okay? Please take note of that. 1 will be released, 5 will be utilized for the regeneration process. Okay, so that's the case when it comes to the step 3. Okay, so now for the last step, which is known as the regeneration. Remember that we are going to stock G3Ps. Okay, we're going to stock 1 G3P, which is a 3-carbon compound. Okay, we're going to stock them. Then, in this particular process, we are now going to utilize 5 G3Ps. Okay, so this would be the G3Ps that we are going to utilize for this part. Okay, now all of these G3Ps will actually create the RUBP. Now the question is, how many RUBPs will be created? Now remember, G3Ps, they are actually possessing 3 carbon backbones, while RUBP, it is now possessing 5 carbon backbones. For this to happen, adenosine triphosphate will be utilized once again. Okay? Now, if we're going to utilize the G3P that are present here, let's simply consider first this one. Okay? We're going to utilize the carbon that is present here to create the RUBP. Since it is only 3, we are going to capture 2 more carbon backbones. Okay? Since we have here the presence of a phosphate backbone, we are going to utilize this for the RUBP, and the other one will be coming from the adenosine triphosphate. So we already utilized 1 phosphate from the ATP. Remember, RUBP, it is ribulose 1,5-bisphosphate. Okay? So that's the first case. Now, we still have few remaining carbon backbones from our G3P. Now, we're going to utilize 3 of them. Once again. Then, since it is not enough, let's get 2 more carbon backbones. The phosphate that is present here will be utilized. Then 1 phosphate from the ATP will also be utilized. Now, lastly, we have the presence of 2 carbon backbones. Then we now have here the presence of another G3P. We're going to utilize 1 phosphate, and the other phosphate will be utilized from the ATP. Okay? So now, how many excess phosphates are there? There are 2 excess phosphates. These phosphates will now be released on the stroma as well to be utilized by the chloroplast. And we just utilized 3 adenosine triphosphate. We released 2 phosphate intermediates. We released 2 free phosphates, and 3 adenosine diphosphate will simply be created. In the end, we created how many RUBPs? We created 3 RUBPs. And if you can still recall, how many RUBPs did we utilize in the start of the cycle? We utilize also 3 RUBPs. Okay, so that's the thing when it comes to the Calvin cycle. We are just recycling every single one of them. Okay, so now, since we already created and regenerated the RUBP, the question is, what are we going to do when it comes to this released or captured G3Ps? These G3Ps are actually our monomers for the creation of the different types of sugars that are actually present there. Okay? Remember, 1 G3P, it is 3 carbon backbones. But if you want to create glucose, you need to have 2 G3P to create 1 glucose molecule or 1 hexose. Okay, so you can see in this particular case, in every 3 cycles, you are going to release 1 G3P. In every 2 cycles, you are going to release 2 G3Ps and create 1 glucose molecule. So imagine how many times Calvin cycle will happen for us to create a complex carbohydrate.